Okay, I think we'll get started. Welcome. My name is Archie Clutter. I'm Dean of the Agricultural Research Division here at the University of Nebraska. And it's my pleasure to welcome you to this session and panel discussion, close range and remote sensing technologies, mitigating drought stress. Peter McCormick closed his comments this morning by asking the question, how do we feed 10 billion people in 30 years with limited water resources? And I thought that really put a point on what we're faced with. He could have also added the complexities of a changing climate. So huge challenges. There's, I know, general recognition by those at this conference that we'll need tremendous innovation in the development and application of new technologies if we're going to meet that grand challenge. And one critical area of development is that of sensing technologies with intended applications to improve yield and yield stability, reduce inputs, and sustain the environment through greater stress tolerance. So for our session today, we have a great collection of experts working on a diverse range of these applications. Each of these participants is very accomplished and brings unique expertise to the panel. So I'd encourage you to look through the program. There are bios that describe that expertise and those accomplishments for each of the participants. I'll do a quick introduction by name and affiliation so that you know who is who, and then I'll ask them to each come up and give about a 10-minute presentation, and then we can have some discussion. Tala Awada is professor of plant ecophysiology in the School of Natural Resources at Nebraska, and she's also associate dean of the Agricultural Research Division. I have to get my order right here. John Gamon is professor of plant physiology and remote sensing in the School of Natural Resources. Ayse Kilic is professor in the School of Natural Resources and Department of Civil Engineering at Nebraska. Sruti Choudhury is assistant research professor of computer vision in the School of Natural Resources and Department of Computer Science Engineering. Yufeng is a member of our faculty, actually is just promoted associate professor, so that's an update to the program in the Department of Biological Systems Engineering. And Harkamal Walia is associate professor of plant molecular physiology in the Department of Agronomy and Horticulture at UNL. So with that, let's start with Tala. Tala's presentation is adoption of high throughput plant phenomics for assessing drought stress in plants.
Thank you, Archie, and thank you everyone for joining us in this afternoon session. I'm going to just provide a 10-minute short presentation to give you an overview about the phenomic efforts we have here with a couple of examples, and my colleagues will take it from there and they will provide specific examples of the research that they have been conducting in the area of remote sensing and high throughput phenomics. So to start with, all right, for a few of you who haven't worked in this area, we have all been doing plant phenomics for millennia, really. 2,000 and 3,000 years ago, we're looking at how the plants look like and what it's doing. The issue, it has been laborious, it has been expensive, and it has been destructive in many cases. So high throughput phenomics is the idea of how we can use images to extract meaningful biophysical traits and physiological traits from plants so we can help and we can apply it to solve challenging problems in agriculture. And that's what we're going to focus about today. So we've all seen the increase or expected projected increase in population, and we've all seen the charge of change in diets that's associated with this. So the idea of meeting the challenge of 2050 and food security is still there. And if we look at this chart, which shows the progression that we are making, and in this chart they use the total factor productivity, which takes into account efficiency or technical efficiency, not only improvement in the crops, we see that we are still falling behind by the end of the century. And it's not so much, I think this is is there a pointer? It's not so much if we look, here it is. It's not so much in the developed countries where we see it's an averaging, if I see well, 1.51, which is a little bit short of the 1.75 that we are required to reach. It's really in the developing countries where this is going to be a huge challenge. And if we look at it, we made some significant improvements over time. This is the reduction in hunger over time. But if it hits 2015 and you see an increase in food insecurity globally, and look closely at it, it's because of climate change and conflict. And we know how agriculture is also related to conflict. So how can we meet the demand for food under these restraints to accomplish our goals? And here is where plant phenomics plays a role. It is really a tool that we can add to the toolbox to address these food insecurities. I have screen over here. So we can, it can help us with speeding up the breeding process by helping us select traits early on. And instead of taking the average 10 or 14 years to get a new genotype, we are hoping that in 6 or 7 or 8 years we can accomplish this. The other thing, it can help to better understand the connection between the plants and their biotic and abiotic environment, including drought and other stresses. And if we know all this, we can really design and work on precision ag that would be precise, and it uses the large data and the data input to help us better manage our resources. So in essence, we are dealing with the genotype by environment, by management, by socioeconomic aspect of this agriculture. And it's a global effort, really. How many minutes I have? It's really a global effort where we are part of what they call the International Phenomics Network. And it's currently there are 34 members in this network. And we are all working for a common cause to advance the science, advance information sharing, and deal with the bottleneck, which is large data or big data problem. Additionally, if you look at the map, there are several sub or regional networks that we are also part of, like the North American network with the same emphasis. There's European network, Australian network, etc. So there are several efforts globally going on to address this issue and moving the science forward. However, interestingly, if you look at the thousands of papers that have been published recently in plant sciences, we still see only less than 400 total publications that have mentioned the word plant phenomics or high-through phenomics or phenomics in their publications. So it has seen significant increase since the early 90s, but we are ways to go from reaching, there's big potential for growth in this area in terms of publications. All right, so now I'm going to shift a little bit gear and say what we are doing in the University of Nebraska to address this issue. Since 2015, we have invested in several systems ranging from the small plants all the way to the field setting. To the left, you have what we call the Lemnatech HTS system, which is designed for small plants to phenotype them, image them. In the middle one is our control system, which I'm going to show some result for it. And in the far left, there is the SpiderCam field facility, which Dr. Guy is going to emphasize the results from. And these all really complement the existing investments at UNL that's being supported by CalMet, going from the leaf all the way to the satellite imagery. So some of the quick results, this system, I'm not going to speak too much about it. It's across the street. If you're interested in visiting the greenhouse, it's automated greenhouse. Each pot is sitting on an automated carrier, and it goes consecutively into 4 chambers to get 3D images of various characteristics in different wavelength. So some of those indices that we get have been very useful in the field are the shape, the architecture of the plant. And we have seen that for cotton, it works very well, because during drought stress, the first thing that changes is the top architecture. So that is trait that you can actually transfer to the field and relate it to water availability in the soil. Another thing that we have been able to do is the leaf counting and growth of individual leaf. And if you are performing early selection of plant genotypes, this becomes a very useful trait. And we can manage now up to 11 leaves, but and we have here the person who developed, Dr. Choudhury, who developed the system, and it has been very useful in selecting traits early on in the greenhouse. Then there are other useful traits that we are extracting. And for instance, the tilting of the stem. This is very important if you are developing bioenergy crops. When you're changing the ratio of cellulose to lignin, then you change the ability of this crop to stand still, or straight. So we can measure this angle, we can measure the wilting, we can measure the distance between these internodes, and all of those are done through artificial intelligence, neural network, using tremendous number of pictures as training sets, and of course with ground validation. So now we are working also on developing these 3D system. You'll see some floating objects, so it's not perfect yet, but it's the idea of eventually how can we manipulate genes and how can we predict and simulate virtually the growth of a plant as we are manipulating these genes. So it's the future. We are not there yet, but we can now grow plant and model it 3D, not perfectly. And then we can measure things we are doing now is borrowing some indices from the remote sensing groups to actually derive some of the physiologically based traits in plants. Some of them have worked, some of them have not. For instance, here, this is an example of chlorophyll. We tested 50 chlorophyll index, and we managed to have 2 to correlate with destructive and other types of measurements. And they did not work very well under all conditions. Some worked better under drought, some worked better under control. So there's really lots of work to be done when we are trying to use a cross-scale and already developed indices. But it worked very well for plant water content. And this is destructive versus derived from images. And you see great relationship in these maize plants under drought stress and high water levels. It also worked very well with deriving some of the nutrients. And last slide, 0 minutes I have, is why hasn't everyone adopted it yet? It's really an expensive technology. So in a study that was done on breeders, they found that many breeders are waiting. Technologies keep changing, and they're waiting for the technology to get cheaper and the models to be developed before the adoption happens. Thank you so much.
So we have time scheduled at the end for questions and a panel discussion, but assuming that everyone stays close to their time, we probably have a little time at the end of each to see if you have a question. Anything for Tala?
Yes.
Thank you. Pasquale Steduto from FAO. Just one question. I saw that there are all these facilities also in greenhouse. We always have learned that the response of the plant to drought depends very much also on the rate of that drought development intensity velocity and so on. So I was wondering, what is the expectation then from doing certain work of phenotypic analysis in the field as compared to the one that you do in the pots?
Well, that's a very good question. I think that the limitation we have in the phenomics facilities in the greenhouse is the same limitation you would have in any other greenhouse. It is really specifically relating, like you would say, specific trait by isolating the remaining of the environment. In the field, it's that's the real place, but really you have complex interactions happening with the plant. In the greenhouse, the advantage is having only, let's say, water or having only high temperature. Try to understand the expression of these traits before you take it to the field and for further testing. Yeah.
All right, thanks, Tala. I think I'm going to use the same order that I have in my program, and that would be Sruti, Choudhury. So welcome to the podium.
Thank you so much.
The title is Deep Learning for Early Detection and Temporal Propagation of Drought Stress using Hyperspectral Image Analysis. Sruti?
Thank you so much. I'd like to talk on image analysis for structural and physiological plant phenotyping for drought stress mitigation. The figure shows a schematic diagram for plant phenotyping computation framework based on image analysis. Here, the plant images belong to different genotypes, are captured from different types of cameras in multiple modalities like visible light, fluorescent, hyperspectral, infrared, and near infrared. And the plants are exposed to different types of stress conditions, including drought, mineral, thermal, and salt stress. So the plants are imaged from different view angles, and this figure shows the complete phenotyping computation framework based on computer vision based image analysis techniques. The outline of my presentation is drought stress mitigation in high throughput plant phenotyping. I would like to emphasize on two different approaches. The first one is data-driven approach for detecting drought stress in plants using hyperspectral image sequence analysis, where we are going to get physiological phenotypes. And the second method that I would like to emphasize is the drought stress quantification using three-dimensional plant phenotypes, where I am going to focus on the structural phenotypes. Structural phenotypes are phenotypes which are identifying the morphological phenotypes, whereas physiological phenotypes basically identifies the physiological processes in the plants. For the method one, we are going to develop or investigate computer vision-based algorithms to classify the stress and non-stress plant pixels. Development of a drought stress index, that means whether the stress conditions are moderate drought, severe drought, or in extreme drought conditions, and also to identify from a specific drought stress, whether the plant can be recovered to the normal conditions by rewatering or not, and also demonstration of temporal propagation of stress levels using machine learning or deep learning approaches, whether we can early detect these stress conditions early. That early prediction is also another important algorithmic development, and analyze the speed of drought stress recovery. The method that I'm going to present on are based on an experiment on cotton plants, where we have used 20 cotton plants in two different experiments, experiment 1 and experiment 2, and in each experiment there were 10 plants, and also those plants were subjected to drought stress on different days. The segmentation process I have shown here, briefly I'm going to explain it. Basically here we are using the NDVI based segmentation technique, where plant images from two different wavelengths are used, and the image subtraction based on image subtraction, we have also used image enhancement and then found the plant pixels. Now those plant pixels are superimposed on the original plant image to find the pixels that correspond to plant and the pixels that correspond to background. Now the pixels that correspond to plant are used for the reflectance curve generation. What happens here, for each pixel we have generated the reflectance curve for all the wavelengths that are present in our hyperspectral camera. Typically our camera has 550 nanometer to 1700 nanometer wavelengths. So for each plant pixel we have developed the reflectance curve, and that we are calling as hyperpixel. That hyperpixels are actually the input to our machine learning system. We have used unsupervised machine learning to group the similar types of pixels to categorize them either stressed or non-stressed, and here we have explored or investigated different types of clustering algorithms using K-means clustering, DB-scan, hierarchical clustering techniques, and then used a elbow method to determine the number of clusters. Here, the interesting result is that using the elbow method, which is really unsupervised, we got the 4 different distinct types of clusters, and we can identify them as abnormally dry, mild drought, moderate drought, and severe drought conditions. Then we plotted all those average spectra of each cluster, and we got these results. Now here the interesting point to note is that where we are comparing between the stressed and non-stressed cartoon plants in the NIR region, we got significant differences in the spectral reflectance curve. That is actually the motivation for this research, that we are definitely seeing differences in the NIR region of the reflectance curves, and analyzing further we can get the different drought stress levels, and also how we are recovering or the temporal propagation of drought stress can be demonstrated or quantified. This is the result from hierarchical clustering, and here some of the results for temporal propagation on different days, where we have demonstrated the green pixels are the unstressed pixels or subject before the stress was applied, and the red pixels correspond to the stress pixels in the plant. This is the result for a different plant and another plant here. Now here it is basically a short video to show the superimposition or the reverse mapping just for visualization. What happened here, for each cluster we have used different colors and then mapped them back on the original pixels of the image to show how the different types of clusters are changing. That means how the different stress levels are propagating with time. This is a schematic diagram for artificial neural network flow diagram. I am not going into details of that, but the classification result is really inspiring, where control and stress pixels are classified with a performance of, I think, 90% or more than that, but the pixels, the classification results are inspiring here using deep learning. Now HyperFino Tool is the toolbox that we developed from our Plant Vision Initiative research group. It's a toolbox which comprises algorithms for unsupervised and supervised machine learning algorithms, and also designed for the hyperspectral image analysis, where for each pixel it will generate you the reflectance curve and also do the clustering technique and map it back to the original plant image for visualization. The toolbox is still under consideration, but we already got it in a beta level testing. Problem 2 is I am going to talk about the 3D plant model reconstruction so that we can analyze the drought stress using 3D plant phenotypes. Now the question is, why 3D, where it is involving lots of complex computer vision and machine learning and statistical tasks? Here there is a motivation for 3D analysis. The motivation is given here. We know the plants are suffering from self-occlusions and variations in phyllotaxy for light interception. They do rotate in their life cycle, and that changes the size of the convex cell considerably. And as the plant rotates, the self-occlusions and the plant rotations, as it is evident from the figure, the convex cell of the same plant from two different viewpoints are absolutely different. So we are not getting the true measurement. So to overcome the limitations of self-occlusions and phyllotaxy, 3D model reconstruction is necessary. We have developed a new taxonomy for 3D phenotypes. I have just 2 minutes left, so I'm not going over this classification. In case you have any question, please do let me know. The main theme of this phenotyping classification is that we have 3 different types of phenotypes here. Structural, which comprises the morphological structure of the plants. Physiological, which identifies the physiological processes plants and the phenotypes that relate to physiology. And the temporal, that is the most exciting and new innovative approach, where it is based on trajectory or the event based. Now, event based phenotypes identify the important events in a plant life cycle, whether they are dependent on different types of stress. That means whether drought stress delay the emergence of a leaf or germination or the emergence of a fruit or flower. These are the event based phenotypes that we are talking about. We are pleased to inform that University of Nebraska-Lincoln has publicly released 3D plant phenotyping dataset. The dataset description and the how to download is all information is available from our website plantvision.unl.edu, and the dataset has the corn plants from 10 different viewpoints, and this dataset can be publicly used and free to download. This is our camera calibration methods. Some of the videos using our LemnaTech high throughput plant phenotyping system, just the opposite building. And this is how we developed the calibration for the systems, calibration of cameras. That means using a checkerboard from multiple viewpoints. And next is the 3D model reconstruction. Here we use the space carving technique for reconstructing the 3D model of a plant. Some of the holistic phenotypes here, and also we have been able to separate different leaves and the stem to measure the component phenotypes like total number of leaves and the stem height. Now just one comment about holistic and component. Holistic phenotypes are the phenotypes which are computed by considering the whole plant as a single object, whereas the component phenotypes are the phenotypes where individual leaves and stem or individual components of the plants like stem, leaf or flower or fruit are considered. And here we have been able to count the number of leaves, the stem height, the stem cross-sectional area, and also the leaf curvature and leaf length. Finally, I would like to acknowledge our team members, Plant Vision Initiative. Here, basically, my team is based on the master students of Computer Science Engineering Department. I am thankful to Tala Awada for her continuous support and also encouragement and educating me about plants, because my background is totally in computer science and computer vision with very limited knowledge in plant science. And finally, thank you very much for your kind attention.
Any initial questions for Sruti? All right, thanks again. Our next speaker is Dr. Harkamal Walia, improving drought responsive phenotypic diversity in wheat from wild alleles. Harkamal.
All right, so I'm going to talk about some of the work that we've used, the imaging technologies for wheat. So my lab's interest is in working on climate resilient cereals, mostly rice and wheat. So today I'm going to talk about our work on wheat. So as we know, and I think all of us are aware here, that irrigated agriculture draws on fresh water supply, and about 20% of our cultivated land is irrigated, but it supplies, the benefits of irrigation are obvious because it supplies about 40% of our food resources. What's so particular about the cereals is that 60% of our production in cereals, that's major wheat, rice, maize, depends on irrigation. So one way, clearly there is better management, but another way is to actually make crops more drought resilient. So that's an area that's been of interest for my lab for a while. So we've been interested in finding novel sources that would make wheat more resilient. So just to kind of lay the concept out, we wheat domestication is about 10,000 some years, 10 to 12,000 years ago. And when you look at wild species, if you go out in the nature and you look, and this is true for wheat and other crops, you have a large diversity in the genes. So basically, diversity in genes being differences in sequences. But as humans selected over the civilization through a process called domestication, we ended up with these land races a few centuries ago, a century ago, that are were optimized for those regions where these crops were grown by farmers. And then, with modern plant breeding, or conventional and modern plant breeding, we continue to select further. But each time you select, through a major allele, you tend to decrease the diversity of the responses and the genes that your cultivar would have. So we have really high-performing, uniform-looking plants, but if you go out in the field and you sequence a handful of wild-growing plants, and you have from the your breeding material, the amount of diversity that you would see is quite different. So basically we're losing that diversity. So the question that we wanted to ask is that, could we go back in the wild, knowing that we have really good tools for genomics and very good tools developing, as you saw with Shruti's talk, for phenomics? Could we go back in and revisit some of the questions that traditionally many people have done through breeding and crosses, but with these two new tools? For this, we have a collaboration with our colleagues at the Hebrew University in Israel, and that's also the spot where you have very high diversity in wild emmer wheat. It's one of the ancestors of the pasta or the durum wheat that we grow in modern times. What's specific, what we were looking for is drought tolerance, but the wild emmer that grows out within very hot and dry environments, it's salt tolerant, it can make a lot of biomass if given enough moisture, and it has disease resistance and other traits that people would want to incorporate into their modern breeding lines. So my colleague made some crosses. So he took the best, one of the high-demanding, high-producing pasta wheat. If you've had Barilla pasta, it's commonly available. So it's coming from a variety called Svevo. And he took one of the lines from the Golan Heights, or one of the plants called Zavitan. Zavitan is named after the stream that flows into the Sea of Galilee. So just to give you an historical context. And then he crossed and crossed until he could get to a point where he had small pieces of this wild emmer wheat incorporated in this high-performing pasta wheat. So that's when we started talking about this project. So he sent a very talented student who's co-advised by me and him from Hebrew University, and asked if we could actually explore this material that he had generated with our phenomics technologies. So this is the setup of the drought experiment. So we had plants that were grown until they were like about 11 days old, and then the control plants, which are called well-watered, were kept at 80% field capacity, and then we gradually decreased the amount of water to 30% field capacity, and then we imaged them starting from here, from the initiation of the stress. So the cool thing about the system is that we can measure very precisely the amount of time, amount of water the plant takes up every day, and then by using imaging, we are able to predict with very high correlation the amount of biomass it's gaining every day. So that results in what, kind of in several indexes, so we could measure how big, wide the plant is, but more importantly, we could ask things such as, when we were about the projected shoot area, which is the estimated biomass based on just the images. But what we do here is I'm going to use a couple of indexes. We have the water use efficiency, which basically says for any given any of the 50 lines that we screened, what was the difference from a daily basis in terms of the biomass accumulation, and then divided by the water use. And then you could do a stress index where you could do that for a control plant and then average it over the entire 50 set of lines. So that gives us a stress index for the water use. So these are some of the plants from Svevo. So if you look at this plant here, and then these are all well-watered plants, but then you look at Svevo and water limited, so you see quite a striking decrease in the biomass for many of the genotypes that we call near isogenic lines. So this is at the end of the experiment, but the key here is how is the dynamics of water use and the biomass accumulation for all of these diverse lines changing every day. Because if you could think of this in one way, like you could have two people who are 6 feet high, and if you were to, at 25 years old, right? But if you were to measure their height every year for 25 years, their height would not match properly, perfectly, even though they end up with the final same product, like, same height, phenotype. So the genetics underlying that of what determines height when you are 5 years old could be slightly different from the genetics or the genes that underlie your height at 12 years or 15 years old. So that's the general idea. We want to dissect that and in terms of drought tolerance for these diverse lines. So this is the heat map. It's very complicated. Given the shortage in time, you just have to take my word that when we cluster these, there's numbers behind each of that color. But if you cluster these genotypes based on different traits, you end up with a cluster of traits that are very stable, which means they don't change that much in response to drought. There's genotypes that are very plastic, which means that they have a dramatic change in response to drought. And then genotypes that have moderate susceptibility, they actually show quite a bit of decrease. And then there's some plants that are just short. And then there's some plants that are very susceptible. So they clustered, this is unsupervised, and they clustered quite nicely. So we decided to pick a few of those from the high stability and high plasticity, and we pursued those. So these are 4 lines. One of them is the parent, which is your regular pasta wheat, and these are the nils. So this is very highly plastic. So if you have high, in dark blue is well watered, but then you see quite a dramatic drop. And these are 2 very stable lines. They don't show a drop until much later, so around 25 days, as opposed to around 10 or 11 days. So basically, we decided that we would, since we can't focus on every line, we decided to focus on one of these. So this is also the assimilation rate. So it just corresponds to a physiological parameter that corresponds with the gain in biomass. What we found was this line 20-2 had a very high root plasticity. So it's quite big when it's drought stressed. But if you look at the roots, it becomes quite small compared to this control condition here under water limited condition, but the roots are quite big. So that showed us that since drought, the roots are the first interface for water limitation for crops. So it was very intriguing for us, so we pursued that. And we looked at the roots in a more controlled greenhouse, long pipe experiments. So you could see under well-watered conditions, on the left are the pasta wheat, and then on the right are that specific line. But however, if you drought stress them, the pasta wheat Svevo drops quite a bit in terms of root biomass, but not so much in the nil 20-2. So that the numbers are kind of shown here in this graph. What we did next was we took even more cleaner roots at early seedling stage, and we did what people call transcriptomics or gene expression. And what we were able to show is that many of the genes that we had already mapped were parts that were different, that were the parts that were brought in from the wild wheat. Many of the genes were mapping there. And in fact, one of the genes that we think maps quite well is shown here to be only expressed mostly in the roots. So the red here represents the part where it's mostly expressed. So we think that we may have a gene that works, that may be responsible for this root response. Of course, a question was raised about, you see these things in pots, but what do you see in the field plots? So what we did was we took a next step where we had a ground bed experiment where, there's about 3.5 feet of dirt in a greenhouse, and we were able to grow these lines, the commercial line as well as this introgression line under different conditions. So we could actually see quite nicely the line that was showing more vigor under well-watered conditions, for instance, is actually showing more pixels. We looked at the assimilation rate. So this is a paper that was published several years ago showing the Zavitan and the Suevo, which are the wild and the cultivated lines. And this is a line that's still higher assimilation rate, which corresponds to the amount of carbon that gets assimilated to increase biomass. So to summarize, we've shown that you could take, there's a very nice fancy pictures, but you can actually derive very quantitative, sensitive data, and you can rederive over several steps to fine-tune your phenotypes. And then the higher resolution your phenotype is, greater is your probability of linking corresponding genes underneath it. So, at some point, they converge, the genomics and the phenomics converges, and that enables us to then go do very detailed molecular and physiological analysis. We also show that there's a value in getting all these wild wheats or other crops into the domesticated germplasm, so you could improve their resilience to heat and drought and salinity. So with that, I would like to acknowledge people who have done a lot of the work. There's Hongfeng's group in computer science and bioinformatics with Shizang and his group, and Yufeng's been helping us with the canopy level, and you will see why we go to him when he presents. And then the collaborators at Hebrew University and lots of genomics resources from the Tel Aviv University. With that, thank you.
I think for the sake of time, we'll move forward. Thanks, Harkamal. Dr. John Gamon is the next speaker. Solar-induced chlorophyll fluorescence as a tool for predicting drought stress in plants. John?
Thanks, Archie. Well, if the lights weren't so blinding, I could see your faces. But so that's similar to the problem that we face when we try and measure chlorophyll fluorescence. If you look at this plant, which you've seen today a few times, it's in the sun, and what you can't see because of all that sunlight is it's actually glowing red in the sunlight, and that red signal is a measure of photosynthetic activity. That's what I'm going to talk about today, and the possibility of using that under stress to detect stress. So we've known about chlorophyll fluorescence for many, many decades. Some seminal work published in the 1940s, for example. What is new is a few years ago, completely by accident, satellites that were developed for global monitoring of atmospheric gases happen to have this high spectral resolution necessary to see a chlorophyll fluorescence signal. The problem is, how do you pick out that red needle in the larger haystack of the sunlight? So if you look at the spectrum in the bottom left, what you see is an example of a high resolution spectrometer. The wavelength is on the x-axis and the signal on the y-axis. And in that well that's at about 760 nanometers is a chlorophyll fluorescence signal. And if you have high resolution spectroscopy with atmospheric bands, absorption bands from the Earth's atmosphere and the sun's atmosphere, you can look in those bands to see this otherwise invisible signal. So from this work, you started to see maps like the one I'm showing you here by Guanter et al., published fairly recently, 2014. We're starting to see the photosynthetic activity of the planet in near real time, only the problem is to get enough signal, you have to integrate these satellite data sets over many days and over large areas. So it's a kind of aggregation of the signal. So there's a lot of questions about what does this really mean. If you'll look at this, you'll see Nebraska is on the edge of a hotspot, the global SIF hotspot. Solar-induced fluorescence is what we're talking about. SIF is the acronym. And so this work is really about work, what I'm going to talk about today is work done at NREC, a UNL facility near Mead, Nebraska. This work we're also doing at other sites to compare other types of vegetation, but I'll just focus on the crop situation, part of this corn and soybean belt, which is the hot spot of the world in terms of the SIF signal. So very high productivity. Of course, also subject to drought. So what do? Well, we use everything from satellite data to aircraft data to measurements on the ground. And our gold standard here is often measuring the crop yield or measuring the actual gas exchange using flux towers, a Ameriflux-sponsored site here at NREC. Andy Suker's in running that. But you'll see a bunch of optical sensors placed in the foreground, including several different institutions' fluorescence sensors. We're trying to compare fluorescence at all these different scales to make sure we're understanding the signal. And you'll see some portable systems as well. So a wide range of technologies being tested here to see what does this fluorescence signal really tell us. Well, what it tells us, what we do know from plant physiology from many, many decades of research, is when the sunlight is absorbed by pigments, chlorophyll, for example, normally for productive photosynthesis, it's used to drive the carboxylation, the uptake of CO2, and the creation of carbohydrates, shown in the box in the right. But under stress, when the stomates close under water stress and plants can't get the CO2, that whole process gets shut down. So what you see is protective mechanisms kick in that are shown on the left, the xanthophyll cycle, for example, but also energy distribution as heat, what's called non-photochemical quenching, the red stuff in the bottom. And that's where the fluorescence signal is sensitive to how much chlorophyll is there, the absorbed radiation, that's the asterisk in this diagram. How much green stuff is at a first cut, we're seeing that with fluorescence. The more green stuff, the more fluorescence. The other thing is, as physiology shuts down under stress, then this other mechanism kicks in, that's the double asterisk, and we're seeing the fluorescence decline. The quenching of the fluorescence, non-photochemical quenching is a technical term. So those two things together are what we're measuring in real time at all these different scales. We're actually measuring the photosynthetic activity almost directly with this signal. So this is fundamentally different from other remote sensing approaches. To say the same thing a different way, if you look at the diagram on the left, the solar-induced fluorescence, sun-induced fluorescence, SIF, is primarily a function of the absorbed radiation. Under stress, that curve changes, water stress, for example. So on the right, photosynthesis responds the same way. It's a function of how much energy is being absorbed, but the slope of that relationship is the efficiency, and that efficiency changes under stress. If those two things change together, then SIF, or fluorescence, should be a good indicator of photosynthesis in real time at all these different scales. So that's what we're testing. This is sort of the core hypothesis of a lot of this research. And this is work collected from Mead. And what we see is a similar pattern to the actual hypothesis. On the left, the SIF signal is functioning, it looks very noisy. So does the GPP on the right, the photosynthesis measured with the flux tower. But if you look at these relationships in detail, you see the slope is changing under different conditions. And under stress, that slope is changing. And so when you put the two together, you actually get a fairly clean relationship, turns out to be slightly nonlinear in this case, between solar-induced fluorescence and GPP. So we're actually following the moment-to-moment, day-to-day changes in photosynthetic activity, including the stress effects. So this is a very exciting advance. it behaves as predicted. It's responding both to absorbed radiation, how much green stuff is there and how much sunlight is there, and also to the reduced efficiency under stress. It's capturing all that information and giving us a nice indicator of photosynthesis in real time. Now, the really cool thing is this occurs at different time scales as well as different spatial scales. So on the annual scale on the left, you see the red curve, the SIF curve, the blue curve, the photosynthesis curve. They're following each other day to day rather well. You see some gap early on due to canopy closure, but for the rest of the season, the SIF is following the photosynthesis. By contrast, the NDVI, which is a greenness index commonly used for drought monitoring or modeling photosynthesis, it doesn't capture the high speed, the sort of short-term variation due to stress or sunlight, clouds, those things, it misses all those. So it's a very different kind of index from SIF. On the right, I'm showing you a daily pattern of solar-induced fluorescence and photosynthesis from the Mead work. This is from last year. And it's following it on a daily scale. So at all these different temporal scales, it has a high fidelity for following the actual photosynthetic rate under all these different conditions. So conventional indices like NDVI cannot really do this. They operate on some scales, but not across these time scales. I'll skip that because it's complicated, but it's saying the same point. The aircraft is used largely to generate images, and people often think of as remote sensing as just getting pictures. Well, no, this is spectroscopy, and we can develop pictures and look at it as a picture. But fundamentally, remember, we're using the spectral information on the right to derive the fluorescence signal and how that changes dynamically. And then we create the picture. And so what you see here is a soybean crop from last year, and in the top, the solar-induced fluorescence signal, the red block on the left, and the same thing shown as NDVI on the bottom. And what I want to show here is that the spatial image, when you show this as an image, this spectral information, there's more information in the SIF. You see these subtle differences in the field due to soil texture and moisture conditions, things that you can't really pick up very well with the NDVI. So we think it adds some information that is not present in the greenness index. So take-home messages. Solar-induced fluorescence provides a close measure of photosynthesis, or GPP, gross primary productivity, across time and space from the leaf level up to the globe, from a moment to the monthly scale to the annual scale. SIF offers new ways to detect stresses, including drought stress. We're seeing that effect on photosynthesis in real time. Now, the challenge is to develop operational indicators from this work. This is basic research. There's more work to be done to develop useful technologies around this. And the differences in instruments and retrieval methods that I don't really have time to go into yield different SIF values. The quantitative information we get depends on what kind of instrument you have, what brand, and how you analyze it exactly. So there's a lot of work needed to standardize this methodology. And so that's pretty much the end. I want to acknowledge the support from University of Nebraska. Ag Hall has been great, supporting the airborne research. NREC is great. Also, the facilities at Comet. But we're also getting some funding from the European Space Agency through the photoproxy program. Lots of collaborators. One thing that's great about here is all these wonderful people who contribute. I'm just going to show you their pictures because there's so many of them. I should throw Lycor into the mix because they've been helping us as well. But people from different institutions are coming here to do this work to try and develop these technologies and test these technologies. And I'll leave you with a pretty picture. You can visualize photosynthesis from this kind of work. And here's a picture of relative photosynthetic rates. This is done for functional biodiversity studies, but it's not that different from, say, phenotyping. We could apply this, in this case, to a prairie outdoors, but you could apply the same concept to phenotyping to look at photosynthesis in real time. Thanks very much.
Thanks, John. Any burning questions for John? George?
Why SIF is better than PAR?
Well, it's got that extra effect. Okay, I'm going to take you to this slide here, or this one here. They're the same thing. This is the concept. This is the data. PAR by itself, or absorbed radiation, tells you part of what you need to know. It doesn't tell you how the plant is responding to that. And that CIF information includes that information as well as the APAR information. They're wrapped together. So this is sort of the hypothesis. It looks like it's working according to the hypothesis, and we get a nice relationship here. Now, if you just followed PAR, you wouldn't see the plant response to PAR changing in real time.
To do APAR, you would. The less sun, the less photosynthesis.
Right. But then there's the additional stress effect, the non-photochemical quenching, which kicks in. That was in that diagram I showed you before. So there's those two factors working together, and being able to see them together is what's unique, I think, about CIF, as opposed to some other methods.
Thanks.
So, thanks.
Thanks, John. All right, Dr. Yufeng, I believe, is next. Yeah, Yufeng. Sensing and modeling of plot scale crop water evaporation at very high temporal resolution. Yufeng?
Good afternoon. So I'm an engineer. So my role here is mainly to develop methods and instruments and systems so that these stuff can be used by the breeders and the crop physiologists and even computer scientists to do the work. So I'm going to talk about phenotyping for water use related crop traits at a high spatial and a temporal resolution. So we all know that water-related crop traits are very difficult to measure. And by crop-related traits, I mean indices such as water use efficiency or evapotranspiration. But they are very important. They're super important for crop improvement in the context of plant breeding, and also for the water and irrigation management in the context of production. So I'm going to talk about that, and I'm going to use two examples that from our phenotyping facility, sort of plant the seeds to show you the potential of what we can do in these very cutting-edge research activities or directions. So many of you already see this slide. So this is our high-throughput plant phenotyping greenhouse, which is equipped with a state-of-the-art imaging modules, and I listed all those imaging systems and the potential traits it can measure. But one thing that is not emphasized enough, but Harkamal did mention that a little bit, is which is shown here in the animation on the bottom right, and it is an automated water station, right? So the plants are coming out of the imaging system and then was transported to a water station, which you prescribe the amount of the water that you need to use, and then also weigh the parts, meaning that you know the exact weight before and after the imaging of that particular day. That really creates the opportunity for us to track what is the amount of water being consumed through evapotranspiration during the day and across the entire life cycle of the crop. So you'll see the animation, so I'm gonna skip that. So here is an example of what we are doing with the data set. Here I show, let me see, here I am showing two plants. So the one on the left is under the normal well-watered condition. The one on the right is under the drought stress condition. Now I put this, this is an experiment over the day of 20 days from the beginning, and we take images once a day, and then we do the image processing of that, and we extract the pixels that gives us the estimated plant biomass, which is plotted on the left. And also, because of that precision water staging, what we can do is to estimate the amount of water use or evapotranspiration, which is plotted as a function of day on the right. And you can see that the blue curve is representing the well-watered plants, and the red curve is representing the droughted plants. Now, a couple of things I wanted to kind of raise your attention here is, so we know that the drought is stressed on day 6. And you can see from the left curve that you actually didn't see much difference regarding the biomass starting that day. So really the biomass accumulation starts to separate probably around day 15 or 16. But if you look at the number of the amount of water being transpired or evapotranspirated, it actually starts from day 6. So when we see this kind of result, it's really surprising that you don't really see an effect yet on the biomass accumulation, but the plants actually start to do something trying to conserve the water, so to speak. So this is, and also, through the tracking, I listed some of the numbers from those 2 plants, right? So for well-watered plant, so on the left again, so the fresh weight is at the end of the experiment, day 21, that is a 91.8 gram, and the dry weight estimated is a 15 gram, and the total water use over that period, 20 days, that's about 1,844 gram. So and then I can use the dry weight and the total water use, and then I can calculate the water use efficiency in terms of dry biomass accumulation, 1,000 gram of water consumed. So that amounts to 8.1 gram of dry water per 1,000 gram of water used. And I can do the same thing for the droughted plant, and you can see all the indices there, and we can do the comparison. Now, one thing I wanted to mention is I show you an example of one plant, but the system that we have actually got the capacity to do this over hundreds of plants over the course. So that was a really powerful analysis that you can do relates to the water use and the water use efficiency. And then the second example that I want to show you is this new spider chem field phenotyping facility, which is situated about 40 miles north of here in the Mead. So this is a cable robot phenotyping concept that you got 4 poles on the corner, and then there's a sensing platform in the middle, which is attached to 4 cables, and then by pulling those 4 cables, you can very precisely to position the sensor on any location in the plot, and then take the measurement. And on the right here is our sensor platform, and you can see that at the bottom of that sensor platform, we got a suite of sensing devices, including RGB, NIR camera, multispectral camera, we got a thermal infrared camera, we got a spectrometers, and we also got a LiDAR scanning system. So here is an example of that one. You may want to back out and to show the original video. So many of you from Nebraska has probably already seen this many times, but if you are first time in Nebraska, this is how the new spider cam system is operated. It's pretty cool. And you will see why it is called a spider cam. It literally looks like a Spider-Man. So you can see the dolly in the middle. So that was hovering on the top of the crop to take the measurement. And it is very precise in positioning, and it's also very agile. That you can see it moves really fast, and then it can go over hundreds of plots multiple times during a day, which is very important for phenotyping because a very bigger hole in the phenotyping is you will not be able to kind of take multiple measurements during a day, that being able to see what's the response in the morning versus midday versus in the afternoon. So you can see it's moving a couple of times across the field during the day. And this is a very typical operation day that we certainly generated a lot of data, and we got a multi-model sensing systems to capture various different aspects of the crop. What I'm going to show you today relates to the thermal infrared imaging from the spider cam system. So this is a time series of, so I'm showing you here is a one soybean plot. So it is a pseudo colored image of thermal infrared image. So the blue means cool temperature and red means higher temperature. What you see here is the two soybean rows, basically horizontally. And the image, so we use a spider cam system and take the image 12 times during the day. And the system gets the capability to measure many plots like this way during a day. And you can see that it starts from about midnoon, 12:30, and it ends 5:20 in the afternoon. And you can see there's so many dynamics and the difference of the temperature distribution in the crop and in the soil, right? So point-based IRT sensors are usually used quite commonly now for the irrigation management, and as well as in remote sensing, they use the thermal band of the imagery to model the energy balance and the evapotranspiration. Now, this kind of information has never been will not be able to create this kind of information yet with other systems. So this is only the first time that we're able to capture the temperature distribution of a plot with very high spatial resolution, as well as very high temporal resolution. And when this is very useful for the folks like Dr. Christopher Neal to sort of being able to separate the surface temperature between crop and the soil and do better modeling regarding the energy balance and the evapotranspiration from the surface. So my last slide here. So this is just sort of I do the image processing of the one that I previously showed, and this is a combination of 36 plots during the day. And what you see here is we can separate the soil temperature, which is orange points, from the vegetation temperature, which is green points, and then you have the blue points, that is the incoming solar radiation. And you can see there's a lot of dynamics between them, and you can also show that if you wanted to use crop surface temperature as indices for drought or something, and the moment that you take the measurement actually matters, right? So you can see there's sometimes they are about the same, but sometimes there's a huge difference between them. So again, it's this is otherwise not possible, but it's only possible with the spider cam system. I'll just throw in there, and there's a lot of things that we will continue to do with this kind of state-of-art facility. And with that, I will close my presentation.
Thank you. Thanks, Yufeng. Dr. Ayse Kilic will finish this up. Oh, sorry. Yeah, go ahead. Sure.
Were you able to separate evaporation from the soil surface from transpiration to the plant when you did the water use efficiency calculations? How did you do that?
So are you, yes, so are you asking about the greenhouse or in the field? Yes, so right now what you see here, that is the evapotranspiration. So we do not, I do not separate them out, but there is a way to do it in the greenhouse. Essentially what you can do is we can intentionally set up empty pots in the greenhouse, and use that as a proxy and monitor the water use of those empty pots to estimate the evaporation part.
Thanks. Ayse's presentation. Oh, sorry. Do you have another one? Sure.
Just for the pot experiment, for the water use efficiency, the dry matter that you have reported is for the whole plant or only for the above, let's say, pot part?
For this one, it's only for the above ground biomass.
Okay.
Well, good afternoon. My name is Ayse Kilic, and I'm going to talk at least a couple of applications that we've been developing over the years. The first one is the EE flux, that stands for the Earth Engine Energy Flux. It runs on Google Earth Engine operating system and uses one of the most widely used energy balance models such as metric, and the primary input that goes into this EE flux is Landsat imagery. Again, the ET, just in case, is the water consumption. Here, as you know, ET varies in space and time. Here's monthly ET maps that we develop for our natural resources district, the Central Platte. And, just as you appreciate, there's variation from field to field. The ET varies because the irrigation amounts are different, and because of all other differences, in management practices. And our natural resources district really appreciate these large amounts of spatial information. Being able to see the water consumption at the 30-meter scale is very important. Again, the beauty is having this on a very large scale for entire natural resources district. And the EE flux is an adaptation of the metric model, and both EE flux and metric uses thermal imagery from Landsat satellite, and to calculate the land surface energy balance components. And here's what you're seeing is the monthly animation of reference ET from gridded weather data. Again, this is running on Google Earth Engine system. And in later July or August, you can see that the mega drought is advancing towards Midwest, including Nebraska. Let me just take, again, this reference ET is sort of potential ET, and that is being built in and calculated from gridded weather system, weather data within the Earth Engine system. Here, let me take you some of the products that's available from EE Flux. Again, the EE Flux runs for entire Landsat archive. Again, resolution is 30 meter, and provides several layers of information, for the users. Like here, what we're seeing is just a true color composite. And for example, albedo, that's important for estimating, the surface energy balance component, and that's just one of them. And the user can download any of these readily available products. You can run one image at a time. That's about 100 mile in size. Again, the image is available every 16 days. The archive goes back to 1985. Again, that was the first time we have the thermal imagery from the Landsat satellite. Here's an application over north central New Mexico. You can see, again, albedo, we have the color doesn't really show up here. The darker color, reddish, is associated with low albedo. We have, I guess this is the pointer, right? Then I need to point it over here. So over the, like the Sandia Mountains, we have low albedo, and then, just high albedo pretty much everywhere else where we have the desert and sagebrush. This is an important product that we derive from EE Flux. It's a surface temperature that really shows us the impact of the evaporative cooling. Again, the same area, just zoom in and look at the details. Again, we have the forest with cooler temperature. Over right, you see those center pivots. Again, cool temperature because of the evaporative cooling. River, the Rio Grande on the left is cooler, so we can really pick up the temperature from the Lancet and make this available in seconds for the area. Again, in addition to several other products, really what is the most crucial product that provides the efflux provides them actual ET and relative ET. Relative ET here we're expressing as the ratio of actual ET to reference ET. Again, we have higher rates of ET over the invasive, over the riparian system, and higher ET up in the forest in higher elevation, again, higher ET rate on irrigated lands. So that's pretty much the application that we do globally, and the data is available free for everybody, again, and every 16 days that is available. The other application that we do with both metric and EE flux is the running these land surface energy balance models at the really 10 centimeter scale. Here's you're looking at an olive orchard in Chile. We work with Dr. Samuel Ortega, one of our collaborators from University of Talca. And the green, you can see the higher transpiration rate from the canopy, and where we have the soil, the red, it's almost bone dry. There is no evaporation, or just a little evaporation. So that is, we haven't really applied it, but it's been going on in South America, and we've been trying to work with Gala, the winery industry in California, around this system with a high resolution. All right, so that was the efflux that provides consumptive water use. The second application that is, again, built in and run on Google Earth Engine is our residential water use. Again, the whole idea is really make use of remote sensing in our natural resources. This is Gear Up, stands for Google Earth Engine app for residential water use and preservation. Again, the ultimate purpose here is really the water management and water saving. And we really wanted to reach out to even a 90-year-old lady who can use some help for, scheduling irrigation for their residential lawn. Really, the problem is, we all have this irrigation sprinkler system, but many of us are scared to death to adjust the controller. Let me ask, how many of you in audiences are comfortable adjusting your irrigation controller for your lawn? Can we raise our hands? Probably this is the most sophisticated. So, a lot of us are really scared to that. It just causes a lot of anxiety. And that's why we wanted to, develop this app, so that, this causes less anxiety. All right, so this is the app. Again, it runs for entire continental U.S. The primary data that goes into this Gear Up is the NAPE imagery. Why NAPE? Well, because NAPE is at 1 meter resolution. It's really small that we can really see those, the homeowner's property, the trees, the lawn, everything. And the dilemma, and again, it has the RGB plus near infrared bands that allows us to estimate NDVI. Again, the nice thing is also it's available. Pretty much every state is mandated to provide, to collect this NAPE imagery. Some states, we have them every 2 years. Here you're looking at a Lancet scene in about 100 by 100 mile. Again, the dilemma with the NAPE imagery was it comes as digital numbers. The data has been collected over several of those private companies, but they just give you a digital number. They do not give a true reflectance, really the physical, the parameter, the product. That's why we also use Lancet as a second thing that we take advantage of this Lancet satellite is to calibrate this NAPE imagery. Here you're looking at the data flow. In addition to this NAEP Landsat, we use soil, vegetation, and lastly, the weather information. The app, again, it runs for 2 weeks from the time you operate, uses past week's weather from RTMA, and next week's weather from the NDFD, the National Digital Forecast Weather Data. Again, we use that to calculate reference CT and also the precipitation that goes into soil-water balance. This runs a complete soil-water balance, and then the model is running on a 1 meter scale. Here, you're looking at home property parcel, an outline in blue that happens to be my house, and I have a huge lot. It's about half acre that I have to mow, and I gotta pay close attention how much water I put, because it's costly in summertime. There were months I paid $600 for a water bill. That's kind of how can I optimize, still keep my grass green, but make sure that I don't pay so much for water. And then this is the NAEP again. Kind of you can see the home and the trees, the lawn. That NAEP imagery, after calibrated with Landsat, is being converted into false color. So here's the classification that we run internally. Again, it's built in, done it on the fly as you run the model, and it makes a supervised classification. Like the yellow is the driveway, and then the home property. You can see the shadow. Shadow is one of the classification, the trees and the lawn. And here's the NDVI, that's it also calculated at the 1 meter resolution. Again, these are not those Google Earth Google Earth images that they are like 5-10 centimeter. This is 1 meter, so it's not perfectly bright and really beautiful. So all this NDVI and other stuff is being translated into what we call is a landscape coefficient, just like a crop coefficient. And for the homeowner's property, this is multiplied with the reference CT, remember, from 2 different weather provider, and it's being converted into the actual water consumption. So again, the whole idea is to provide something so simple. Here, like the forecast, the orange line is the consumption, the water consumption for past week and for the next week, and the weather forecast, the precipitation is with the blue bar. So we provide, that's the first information. Second information, again, running this daily weather water balance model. We have irrigation scheduling. Again, all we want to know is binary code, yes or no, and which day we want to irrigate. Just to simplify the amount of information that a homeowner must enter on their smartphone or iPad. So at the bottom, depending on the sprinkler's discharge, you can choose how, again, time, that's the simple, that's how human mind can understand easily. How long do I need to run my system, or zone 8 or zone 6? So those are very simple information that we develop. The last work is the real time on the fly. How am I doing with timing? Okay. Okay, so the last work that we would like to, and we've been kind of working on it, is to develop an on-the-fly irrigation scheduling in agriculture for U.S. or for continental scale. It's called ETM, is evapotranspiration management and replacement tool. Again, it's gonna be built on EE flux and gear up, pretty much the things that we developed. The primary motivation is really make this Landsat imagery available to farmers' smartphone. You know, the scientists, the federal, the state employee, the private companies, they all have access to this freely available 30-meter resolution Landsat imagery. Why don't we make them available to our poor farmers or rich farmers? It doesn't matter. Why can't they see their field, you know, every 2 weeks when this image is available at the 30-meter? Why can't they look at their farm? So that is the motivation, really make this Landsat imagery available on their on farmer's smartphone. Also, a simple, again, binary, irrigate or not irrigate. Just develop this simple tool. Again, with a unit that the farmers understand. How long do I need to run my center pivot system? And so that's the same idea. Again, you know, make this imagery available to the farmers. That's the, and then especially for, you know, smartphone application, and provide irrigation scheduling. Just like how long do they need to, when they need to irrigate. And lastly, I would like to leave you with a concept of having daily Landsat imagery of Earth selfie. Don't you want to have a Earth selfie, you know, every day? Don't we want to, you know, we have these smartphones, iPhone, we take our selfies. Why don't we take a selfie of our Earth every day? But the problem is we have only 2 satellites up there operating, and we need 16 of them. So we got to put a lot of pressure to our Congress, our federal and state, you know, people, you know, make sure that we have the resources going to these satellites. And you know what? It only costs $3 latte per American per year to have 16 Landsat satellites operating, so that we have our selfie of our Earth selfie. Thank you.
So we have a few minutes. Why don't we just go to any questions for any of the speakers or for the panel?
As an Italian, I had a question for the Barilla pasta wheat. No, my question was, you have done pot experiments over an early stage. So I see two risks there. One is the fact that the dynamic of the drought development in the pots is different from the one in the field. And the second is that you are extrapolating that what happened as a response to drought in early stage is also for the rest of the different phases, which being a complex response, I mean, is a risky business. How do you see this or manage this risk?
I think that's a good question. The way the material was developed was my collaborator had about 190 some lines that he tested for 2 seasons in hot and dry and hot and irrigated environment in Israel. And then from the ones that looked comparable in height and flowering time, because as you know, in the Mediterranean, there's always a terminal drought when you're growing wheat, that the flowering time, if it gets too late, in very good environment, you might get higher yield, but most of the time there's a penalty because it's going to get hotter and hotter. So he selected a set of lines that looked agronomically, in terms of flowering and height, so there's not many issues with management. A subset of 51 or 54 that he sent to Nebraska. Then once we did the experiments in pot, I think I kind of rushed through that, but we did have a greenhouse set up where we were growing these plants at the commercial seeding distance and spacing to again start to translate back from a pot to the ground bed. So it's a canopy measurements. And then I think the students now going back and making some more back crosses to make those pieces of wild wheat smaller and smaller and the background cleaner. So it's very similar to the Barilla or the Svevo variety, but just the small genetic piece from the wild, and they're testing it again in the field. So I think it's a what's that? So for the line that we chose, I didn't have time to show you slides, but if I can show you in my computer if you're interested. But the biomass increase that you see with the introgressional line gives you more robustness during the limited water condition in the ground bed here, but the yield is very similar. What's different is the length of the spike, because these features are not controlled by one gene or 3 genes. It would be great if they were, because then we could just quickly find them. So these are complex traits that are controlled by many features, so by many genes. So what we did find was that the spike length, which typically not always would correlate with the seed number, that was bigger in this introgression line. But other introgression lines that I didn't have the time to show you the data, and we didn't go in great detail, do have differences in yield, but they also have other issues such as being very tall and so on. So there's
Well, I think we're at our appointed time. I think you'll agree with me, some really interesting work and applications. So please join me in thanking the panel.